Okay, thanks Antoine, and thanks for the organizers for organizing such an amazing meeting, and for inviting me to present some of our latest work. And Antoine had to mention the Ambo Young investigators because they paid for my trip. Okay, so in our lab we're really interested in trying to understand transcription from a single cell and a single molecule perspective. And if we think about transcription, it's a highly regulated process, and it's essential for multicellular life, where every cell in your body has the same DNA, yet different cells have different functions and shapes because they express different proteins, and this is mostly regulated at the level of transcription. And so when we think about this, we often think, oh, if a gene is being expressed in a certain cell type, then this must continuously be produced. But in fact, we and many other labs have seen this transcription is actually a highly discontinuous process, and it occurs in bursts. So what I mean with this is that often when a transcription is on, several polymerases initiate close together, and this is then followed by a period where transcription is silent, which can last for hours or even up to days. And I hope you can see this also in this video, where we labeled the transcription of a single gene, and every time you see a spot on a nucleus, this is when this gene is being transcribed, and you can see that sometimes the spot is quite bright, but that also disappears, so transcription turns off. And this transcriptional bursting is a conserved property that occurs from bacteria to budding yeast to human, yet we still have a very limited understanding on the origin of mechanism of bursting. And so in our lab in the last 5-6 years or so, we tried to understand the molecular mechanism of this bursting and how cells switch from this active state to this inactive state and back. And to do this, we used this live cell imaging approach that was developed by Rob Singer's lab, where you introduce repeat sequences called PP7 or MS2 into an endogenous gene. And when these are transcribed, they form hairpin loops. And then we co-express in the cell a co-protein that specifically binds to these hairpins that is fused to a fluorophore. And that results in the appearance of a bright spot in the nucleus, of which the intensity is proportional to the number of RNA that's being produced. So we can now follow transcription as it occurs in live cells. And in our lab, we now do this live cell imaging, both in budding yeast and in mouse and human cell lines. But for today, I will mostly focus on budding yeast. So with this method, we try to address what regulates this transcriptional bursting. And the first thing we thought about is maybe it's regulated by the transcription factor. So as we heard also yesterday, transcription factors are these very important proteins that bind to motif sequences in regulatory regions that are near genes, but they're not static, they're actually highly dynamic, and we wondered how this affects transcriptional fluctuations. So when you visualize transcription factors in live cells, I hope you cannot really see it on this screen, hopefully on some of the other screens, but you can see the transcription factor binding to DNA is actually highly transient, and it actually only seems to bind to DNA for a few seconds. So you should be seeing dots here of individual molecules where you can see when they are less mobile, so we think that they bind DNA, that this actually occurs very shortly. So schematically on a single gene, you might imagine something like this, where transcription factor binds for a few seconds, but it also releases, and so there's long periods where this gene, where there's no binding. And one of the major outstanding questions in the field is how can this very transient binding result in robust transcription activation? And in other words, how can we go from this few second transcription factor binding to this minute or hour-long burst? And how can we connect these different time scales? And one of the reasons that we couldn't really answer the question in the field is because of technical challenges, where we were able to measure transcription factor binding in single cells and transcription in single cells, but combining these techniques have been challenging, so we couldn't really understand what the functional effect of this binding event was on transcription activation. So we now developed a novel technology that allows us to track the transcription site in 3D, and we do this by using an astigmatic lens to determine the Z position, and we use an active feedback loop to keep the transcription site in focus. And then we'll simultaneously measure the binding kinetics of transcription factor molecules to this locus, and then we can see what the functional effect is of this binding on transcription activation in the single cell. And we use the GAL genes and budding yeast as a model system. So we label the transcription of the GAL10 gene. This gene has 4 binding sites, and there's only a few target genes of this transcription factor, GAL4. So we hope that we can see a lot of binding of transcription factors to this locus. And here I'm showing you an example movie where we can track the transcription site inside the cell and at the same time monitor the binding of the GAL4 molecules to this target gene. And so this results in traces such as this, where we can follow the fluctuations in purple of the transcription factor and in green of transcription. And I have to say, in this experiment, we labeled roughly 20% of the GAL4 molecules. So that means that if we see binding of transcription factors, that can mean that there's either one or multiple molecules bound to the locus. But if we don't see binding, that could either mean that there's no binding. It could also mean that there's binding of molecules we haven't actually labeled. And so to analyze this data, we used the hidden Markov model to divide these traces in periods where there's transcription and periods when there's transcription factor binding. And as the first analysis, we took all these transcription factor binding events and we aligned them to the start of the transcription factor binding. And then we averaged the intensity of the traces of the GAL4 and of the GAL10 transcription, and this is then what it looks like. So we see that if we align on the start of transcription factor binding, we see an increase in the signal of the transcription factor, as you expect. But this is rapidly followed by an increase of the transcription activity. And when we do this with higher time resolution, we see that there's a delay of roughly 1 or 2 seconds. But so it seems to be very rapid that when transcription factor binds, we see an increase of transcription. What happens if we align to the end of transcription factor binding, so when the GAL4 dissociates, then we see that this is rapidly followed by a decrease in transcription. And so this suggests that GAL4 needs to be continuously present at this locus to keep transcription going, because when it leaves, it also deactivates transcription. So to look at this another way, we took all the bursts, all the transcription events, and we divided it in 5 time periods. One is before the burst, and at the start of the burst, during the burst, at the end of the burst, and after the burst. And first we just looked at the occupancy of GAL4, and what you can see here, that there's higher occupancy of GAL4 during a burst, so again suggesting you need continuous occupancy of GAL4 to keep this burst going. And that is much higher than before or after the burst, and at the start and end it's kind of intermediate because it's associating and dissociating. When we now look at when GAL4 actually associates in these 5 time periods, and we look at the association frequency, we said that the highest frequency is at the start of the burst. And this is also what you expect, because if GAL4 associates, you see an increase in transcription. So we see an enrichment of association here. But what's very interesting is that we also find a very high association frequency during bursts, suggesting that even when transcription is already on, we often see GAL4 arriving on the locus. When we look at the dissociation frequency, again, as expected, we see a highest frequency at the end of the burst. So when GAL4 leaves, this ends the burst very often. But we also see a very high dissociation frequency, again, during the burst. So GAL4 often leaves when transcription is on. And so this suggests that there may be exchange of molecules during a burst where many come in and then others where molecules leave and others come in, that they can kind of take over and keep transcription going. We then wondered, are there also molecules that basically stay bound throughout the entire burst and can just keep the burst going? So we quantified for each burst what the percentage of overlap is with the transcription factor binding event. And we do find bursts where the complete burst is bound by a single transcription factor, but this is only 16% of all the bursts. So the majority of the bursts actually don't show this complete overlap. And for long bursts, which are only about 3% of the bursts show a complete overlap with the transcription factor, suggesting that longer bursts are particularly important, that there's transcription factor exchange to increase the burst duration. And so to look at this another way, we looked at how many binding events do we see either for short bursts here on the left or for long bursts on the right. And indeed, we see that long bursts show much more binding events and also more than a control gene, suggesting that indeed we see this continuous exchange of multiple molecules binding sequentially to keep the burst going. So overall, our data is consistent with this model where individual transcription factors can bind quite shortly, but then they continuously exchange to keep high promoter occupancy, and this increases the transcriptional burst duration from a short binding event to a long transcriptional burst. And then when all these molecules dissociate and we switch to a low occupancy state, that this turns the gene off and results in the silent period. And this switching between high and low promoter occupancy also suggests that there's some form of cooperative binding, where binding of a transcription factor is more frequent when one molecule is already bound, and so that you actually switch between two states instead of creating a continuum of binding states. And we know that GAL4 actually binds DNA cooperatively, so that was really nicely fit our data. And we think that maybe this model of exchange may be a general mechanism also, for example, for enhancers in more complex eukaryotes, where you have many binding sites, where this continuous binding and high occupancy can result in robust gene activation. So overall, I showed you that the binding dynamics of the transcription factor in fact play a very large role in switching between this bursting behavior and going from high on and off periods. We're now follow up on this work, and I hope you had a chance to see the poster of Wim de Jonge on Monday evening, where he showed that we're going to look at various domains of GAL4, so the DNA binding domain and the intrinsically disordered domains, and how they affect this binding dynamics. We next ask, okay, so we know that transcription factors don't actually act alone, and also, as Nadine already pointed to, there's this clustering of transcription factors, and we also observed this for GAL4. And so we asked, how does this contribute to gene expression, and does this model hold where if you have a transcription factor cluster, can you then quickly recruit multiple polymerases, and then when the cluster dissolves, would it just be the end of the burst? And I don't have time to go into this during my talk, but I really encourage you to go to the poster of Jos Meuse, who's presenting our recent work on this. And just as a very quick summary, we think that these clusters can help to recruit transcription factor to the locus to maybe result in this cooperative binding. But we don't think that the molecules that are in the cluster that are not DNA bound actually contribute to transcription activation. And so they can't really recruit polymerase or the transcriptional machinery. And in fact, they may have a slight inhibitory role on transcription. So what I do want to talk the remaining minutes of my talk about is DNA supercoiling and how this affects transcriptional fluctuations. So as we've heard earlier during the meeting, transcription generates supercoils. When RNA polymerase transcribes through a gene, it generates positive supercoils or overwound DNA ahead of polymerase, and underwound DNA or negative supercoils behind polymerase. And this feeds back on a transcription process where negative supercoils is thought to promote transcription initiation, and positive supercoils is thought to promote transcription elongation. Yet if you have an excess of DNA supercoils, this can also inhibit both of these processes. And so there's various enzymes in the cell called topoisomerases that can quickly relieve these supercoils in order to prevent this transcription inhibition. What we know, however, is that in bacteria, one of these enzymes is present in limiting amount, and so at highly transcribed genes, there's so much buildup of supercoils that cannot be released that this starts to inhibit initiation and elongation, and this turns the gene off, and then you have to wait for release of supercoils for transcription to resume again. So in bacteria, it's thought that this accumulation and release of supercoils is the main driver of this transcriptional bursting. Yet whether this process plays any role in regulating the dynamics of transcription in eukaryotes was so far never been tested. In addition, we know from supercoiling mapping studies that these supercoils can travel from where they're generated, and so potentially, if a gene is transcribed and generates supercoils, these supercoils can travel and influence transcription of a neighboring gene. So we asked, how does DNA supercoiling affect the transcriptional bursting of neighboring genes? And for this, we again went to budding yeast, which have 3 genes of the GAL system, which are in close proximity on the chromosomes, GAL1, GAL10, and GAL7. They share binding sites. They have all binding sites for the transcription factor GAL4. These GAL1 and GAL10 actually share the binding sites in the promoter. And they're in close proximity, so we know these supercoils can travel within, or we think these supercoils can travel within the distance. They're highly transcribed, and also they're in different orientations. So GAL1 and GAL10 are in the divergent orientation, which means we think that supercoils can accumulate maybe here in the middle, and then there's positive supercoils in the gene bodies. But GAL10 and GAL7 are oriented in tandem, which means that the supercoils can alternate. So there's negative, positive, negative, positive. So we wanted to address how supercoils affect transcription of these genes, but we didn't even know what transcription of these genes looked like in wild type, so this is what we started with. And we tagged transcription of first the divergent genes, GAL1 and GAL10, using the MS2 and PP7 system, so we could visualize transcription of both of these two genes in the same single cell. And here I'm showing you an example trace where we can see that, well, maybe not if you're on the front row, but hopefully in the back, that sometimes both of these genes are turned off, sometimes both of these genes are on, and sometimes only one of them is on and the other is off. So to unbiasedly analyze this data, we used a cross-correlation function which correlates these two signals and then shifts all these two signals with a different time delay to see what the correlation is at different time delays, and then you get a cross-correlation function. So this is what the cross-correlation function looks like for GAL1 and GAL10. We see there's a peak at a time delay 0, and this decays to 0 correlation at roughly -100 and +100 seconds, and this is about the time that these genes burst. So what we think is that these genes show correlated initiation, and then they transcribe both these genes, and after that their transcription is uncorrelated. This correlation, or the coupled transcription, only occurs when these genes are in close proximity on the same chromosome, because if we do the same experiment for the same two genes on two different chromosomes, we observe a flat line and no correlation. So perhaps it's not surprising that GAL1 and GAL10 show this coupled transcription because they share a promoter region. But when we did the same experiment for the tandem GAL10 and GAL7 genes, which have separate promoters, we still observe a peak at 0. It's very weak, it's much weaker than for GAL1 and GAL10, but it does suggest that also these two genes show this coupled transcription. So in wild type, initiation of the GAL genes is temporarily coupled. How do DNA supercoils affect this coupling? So to answer this, we conditionally depleted the topoisomerases. Yeast have two that are important for gene regulation, TOP1 and TOP2, that regulate both overwound and underwound DNA. And so what we could do is we added these auxin-inducible degron tags to both these proteins, and then upon the addition of auxin, this resulted in rapid conditional degradation of these genes. And so the system nicely works. If you add auxin, you get very quick degradation of the topoisomerases. However, we did notice that if we didn't add auxin, so we just add all these systems to the cell, we already get some basal degradation of the topoisomerases. So that means that we now have cells with different levels of topoisomerases, with partial and complete depletion, and different levels of supercoiling accumulation. So this is, again, what the wild-type cross-correlation looked like. If we now deplete the topoisomerases, first of all, we see that half the cells don't transcribe anymore, so they turn transcription off. But in the cells where we do see transcription, this is what the cross-correlation looks like. So we still observe a peak at 0, indicating that there is still some coupled transcription, but we now start to see these remarkable values at -100 and +100 time delay. Seconds time delay, yeah. So what this means is that these genes, whenever one of these genes transcribes, let's say GAL1, that it takes roughly 100 seconds to transcribe this gene. It generates so much supercoils at this moment that now this starts to inhibit subsequent transcription of the neighboring GAL10 gene. And the reverse is also true. When GAL10 transcribes, then 100 seconds later, GAL1 is being inhibited. So topoisomerase depletion causes inhibition of transcription at neighboring genes. We see the same for GAL10 and GAL7. And when we now looked at how often do these genes now still co-express, we saw that for both of these gene pairs, the overlap of transcription of these genes was reduced. And this was slightly surprising, especially for the divergent genes, because it had been predicted before that if you have more supercoils for divergent genes, that this should actually enhance the coupling of these genes, because you get more negative supercoils in this promoter region. And so we verified it with another way. We got the same results, and then we started asking, okay, why do we see this decoupling of these genes? And we started to look at the individual transcription levels of these 3 different genes. And this is compared to wild type. So if we partially deplete the topoisomerases, we see that mostly GAL10 goes down in transcription, whereas GAL1 and GAL7 are much less affected. And if we completely deplete topoisomerases, then all 3 genes go down, but still GAL10 is much more affected. And this was quite remarkable because normally GAL1 and GAL10 are always expressed to the same degree. So somehow GAL10 was affected much more here. And we thought maybe that's why we now see this lower coupling of these genes. And we thought maybe this is then because GAL7, there's this highly expressed GAL7 gene, and maybe this is now repressing a transcription of GAL10. And so to test this, we removed the transcription factor binding sites of GAL7, so there shouldn't be any GAL7 transcription. And then we again looked at the correlation of GAL1 and GAL10 with a different method. And you see in wild type, if we deplete GAL7, there's really not much of an effect on the coupling. But if we remove the topoisomerases here in blue, we see a reduction in coupling, and this is almost completely rescued if we remove downstream GAL7 transcription. So this data suggests two different things. When there is not enough topoisomerases, then GAL7 transcription starts to inhibit GAL10 transcription, which then causes the decoupling of GAL1 and GAL10 and their co-expression. But what it also shows is that in wild type, apparently there already are sufficient levels of topoisomerases to prevent this transcription inhibition, because if we remove GAL7 in wild type, nothing really happens. So this data seems to show that these genes inhibit each other and that there's a very strong inhibition of GAL7 transcription on GAL10 transcription, but what now inhibits this transcription so much? So to look at this mechanism of inhibition, we looked at the binding dynamics of the transcription factor GAL4, we tagged it with a halo tag, and we looked at the dwell time of individual transcription factor molecules in live cells, and this is the survival probability of the residence time. In blue here you see wild type, and then when we deplete topoisomerase partially or fully, you see that the residence time of GAL4 is reduced. And you also see that when we quantify the average. So this means that when supercoils accumulate, that there is a destabilization of the transcription factor. And very likely this is then resulting in a decrease of transcription activation. So just to summarize, what we think is that in wild type, the transcription of the GAL genes is temporarily coupled, and you need rapid release of supercoils in order to maintain this coupling. And then when you deplete topoisomerases and supercoils accumulate, then we start to see that all these genes start to inhibit each other, and this is likely caused by a destabilization of the transcription factor binding from the DNA. But what's also interesting is that in wild type, apparently the levels of topoisomerases are high enough to prevent this transcription inhibition. And so this suggests also that probably in wild type, the supercoils and their generation is not the main driver of the transcriptional bursting as we find in bacteria, but that they're actually quickly resolved by the topoisomerases. What's also interesting maybe for people working on other processes is that supercoils are generated by much more processes also during DNA damage repair, and so it's interesting to think about how this could potentially also start to influence transcription of neighboring genes. So just to summarize, I've shown you multiple projects that we're working on, and we think that transcription factor binding dynamics play a major role in determining some of these fluctuations, but there's likely many other processes that influence it, but the DNA supercoiling is probably not the main driver. I hope I have convinced you that using live cell imaging approaches, that we can learn a lot about the mechanism of transcription. And with that, I want to thank the people that did all this work. The first story about GAL4 binding to GAL10 was really driven by Wim Pomp with the help of Jos Meeuwse. And the supercoiling work was mostly done by Hedda Patel with the help of Stefano, Wim, Ineke, and some more. This story was also in collaboration with Domenico Libri's lab. I want to thank my funding, and thank you very much for your attention.
Thanks, Tineke. Questions?
That's super beautiful, thank you. So I was wondering about the cooperative binding of the GAL4, whether your traces would allow you to actually see simultaneous binding, so cooperative in the functional sense that they help each other in time, but also that they help bind actually on the same DNA molecule, and whether that has consequences for the bursting. Can you see two factors binding at the same time from the fluorescence intensities?
Yeah, we would love to see that. We do see that sometimes the intensity of the trace is more than what we expect from a single molecule, so we do think that sometimes 2, 3, or 4 molecules are bound there. But we haven't tried labeling them, for example, with 2 colors. I'm not sure that's whether we will be able to do this. That would be really cool so that we can see it. I thought also maybe we could use the slope to see the quickness of the binding, but I don't know if the data is clean enough to be able to do this. So, but in vitro, there's been really nice experiments where they can see that if you look at the occupancy in vitro of this gene, that there is this cooperative binding where if one binds, then you get binding of all of them, and you need all the regions outside the DNA binding domain to get that cooperativity. So apparently there's these self-interactions, which probably also result in this clustering that can then quickly recruit many of the other molecules.
Hi, very nice talk. Just two questions. The first one is a curiosity about the first part. Since GAL80 modulates GAL4, have you done any experiment to see whether modulating GAL80 you will change the dynamics of GAL4 binding? Because this is something that you would expect.
We haven't looked, I think, at GAL4 bindings per se, because so in these experiments this is full galactose, so we know that GAL80 addition, removal doesn't do much else because it's already fully active. We did look at bursting previously, where we checked what happens if you go to lower galactose concentrations. You mostly see that the burst frequency is reduced, but the burst duration seems quite stable. So if we think that the burst duration is mostly mediated by the GAL4 dwell time overall, like the high occupancy state, then likely this is not so much changed. It's mostly the frequency by which they enter this state that should be affected, but we haven't measured this.
Yeah. And the second part, I mean, these two genes are regulated by the same factors, and as you mentioned, GAL4 dynamics is playing right there.
Yeah.
You have two things going on. Then, would you think this can be extrapolable to two genes that are regulated by different ways, two divergent genes in genes? Because then you would only rely on supercoil, but perhaps the dynamics of the activators is not going to be co-regulated, as it happened in this case.
Right.
And therefore,
so you're saying if you have two different transcription factors, it's probably going to be uncoupled.
Then perhaps maybe.
Yeah, we thought, so in yeast that's difficult to test because it's actually not so easy, I think, to insulate these genes, because if you would have two transcription factors, they would both basically regulate both genes at the same time. In mammalian cells, this is likely true, although there you also have many transcription factors on the same, like on an enhancer region, and potentially they can influence two promoters, right? And they could maybe then simultaneously activate both genes. And now there's this beautiful work in Drosophila that genes are also being coupled there. So it might be a more general phenomenon. But so just to be clear, we don't really know if supercoils play a role in this coupling. We actually think it might mostly inhibit it, and it's mostly generated by the transcription factor binding, I should say.
Yeah. By mammalian may be much more different because genes are far
exactly.
away,
yeah.
and you have to play with enhancers.
Yeah, exactly, and there we also don't know how far the supercoils can spread, so whether supercoils are even, maybe it's completely buffered out by the sequence. This is something that I think is really unknown, which is why we chose yeast, because there we can actually, it's a very clean system. Yeah.
Yes, it seems to me the most of your talk, I think, was a very strong proof that supercoiling is involved in bursting. And then you sort of at the end said, well, but because we find that the cross-correlation of two things that are distant is destroyed by the existence of topoisomerases, that means that it's not involved with bursting at all in the normal case. But I don't see that that's the case, because in one case, the topoisomerase has to fall somewhere between the two genes over a much longer time. In the other case, you're actually talking about the accumulation of the supercoiling in the vicinity of the first gene, and that could be quantitatively quite different, whether there's enough topoisomerase to affect that.
So one of the reasons I say that in wild type we don't think these enzymes are limiting is, for example, when we deplete topoisomerases, we also see that this transcription of one gene starts to inhibit itself. So you see that towards the end of the burst, you get a depletion of initiating again. So in the autocorrelations, we also start to see these dips. We never see them in wild type. We don't see the dips really in wild type, in the cross-correlation functions or in the autocorrelation functions. Only for GAL10 and GAL7, we see a very weak dip at one of the two sites, so it means that probably it's at the edge. And so that's why we say, in addition with the fact that if we make mutants, that they don't really seem to help anything further. So that's why we think probably it's not It might still contribute, but it's not the main driver. We don't see the strong inhibition when you get transcription that the gene actually turns off. We only see that when we deplete the topoisomerases. So they can affect transcription, but in wild type, they're likely not the main thing affecting transcription. Does that make any sense?
Well, it makes sense. I would have just said that I would like to see much more quantitative analysis of the thing than this qualitative statement. I'm not
right,
sure.
But we can discuss.
Okay.
I can show you the paper where we actually quantified all of this, but I didn't have time to show this all.
Yeah, thank you. Very nice talk. Actually, you mentioned very briefly the clustering of GAL4. So do you think that all the GAL4 are bound to DNA in this cluster, or do you think that you have more molecules?
No, we see that there are some DNA-bound molecules and some non-DNA-bound molecules. So if we remove the DNA binding domain of GAL4, we still see clusters. Then when we take and we visualize that, and then we take a wild type that has the DNA binding domain, we see that it can actually go to the ones where it's DNA bound, suggesting that not all the molecules are actually DNA bound.
Okay, so just back to what you just showed. So how do you differentiate the ones that are really bound from the ones that are just like in the cluster?
Exactly, we don't. So we, in this experiment with the first story, we assume that when it's like close, it's bound. That means that we detect a lot of molecules that are not DNA bound, that are close, which would only increase the noise in our system. Yet we still detect some, that's roughly actually 20% of the times we see binding, it increases in transcription. So we do pick up this specific bound molecules that seem to activate, but you're right, many of the ones that we call bound are probably not DNA bound.
Okay, and you don't have any difference in terms of the dwell times for these events that are the ones that are associated with increased transcription versus the others?
That's what we want to test now. So we want to make mutants where we can actually address this question, because right now we don't. Yeah.
Thank you.
Okay, let's thank Tineke again.